Dendritic Chapter slides. All right. Everybody saw how to get to these on Moodle, correct? Okay. Does anybody have any questions about the material that that you guys self-studied on Moodle? So there was a video, there were lecture slides. How did y'all feel about your first self-study day? Could you follow? Was it things that you needed? Now some of you are like, oops, we had a self-study day. I didn't know. Make sure, good, I've got some thumbs up at home. Make sure you do those study days before you come into class. Because I am going to assume if there's no questions that you know all that stuff. And maybe one day I'll throw out a little pop quiz. I don't know. We'll find out. So if I throw out a pop quiz, raise your hand if you think that you can make decent score. Liars. I'm just kidding. I believe you. You say you self-study, I'm going to believe you, so you give me a reason over. Okay? I'm not going to do it. Okay? But know that the offer stands. This is the kind of material that sometimes you don't know what you don't know until you're tested. You're like, I got that. I know that cellular stuff. And then I test you and you're like, I don't got that. I don't know what this is. Okay? So I am more likely to probably throw out a pop quiz just to kind of gauge how we're doing. Okay? So it's not a part of your grade. It's just going to be gauging where we're at. Okay? No pressure. So don't be like, oh my God, next time she's going to quiz us. Maybe, but there's not going to be a lot of pressure associated with it. Okay? I like to use this app called Socrative. Has anybody used this? It's really fun. Okay, it's like polling stuff. So even you guys at home can join us on Socrative. Basically, I'll throw up a quiz, and everybody will answer based off of that. So I might do that next time. I'm not sure. We'll see how today goes. Okay. Another thing too is that I have on the syllabus that we're only going to spend a day on chapter 2. But as I start to look at my slides and really think about how quickly I'm going through them, we might have to bleed into the next class with this stuff. Okay. I want to make sure that you guys have the right foundation so that when we start getting to more complex stuff, you guys know where we're at. Okay. So before we get started, last call. Any questions about the chapter 1 stuff? Everybody good? Okay. So your self-study days will usually have a lecture video like that with some kind of slides and material. This time we just happen to have the first chapter that you guys were able to actually read, since many of you don't have the book due to bookstore issues. So I felt that that was out of your control, so I wanted to give you guys some materials to be able to study. Okay? So is everybody good? Everybody good at home? We can move to chapter 2. Thumbs up. Yep. Thanks, guys. Okay, perfect. Thank you for the thumbs up. All right, so let's go. We're talking about here all about, and I'm going to move this. I might have to move this around a little bit. Actually, I'm going to hide this because you guys don't need to see it up here. Okay. So we're going to talk about the nervous system. We're going to talk about how it's divided up. We're going to talk about the cell types and all of that. But one thing, and we're going to revisit this again later, one thing I want you guys to know is the organization of your nervous system and how it's broken up, okay? So if you see here, the nervous system is broken into 2 major categories. We have the peripheral nervous system, or the PNS, and the central nervous system, or the CNS. The central nervous system is where we're going to spend pretty much solely most of our time in this class. It's everything brain and spinal cord, okay? So we're going to focus the majority of this class on the CNS, okay? But I do want to make sure you guys are aware of how the PNS is. It sounds a whole lot like penis. Did you guys hear that? I feel like, okay, so I'm going to call it peripheral nervous system because it really sounds like penis. I'm going to talk about it, but in reference to the central nervous system, okay? So the peripheral nervous system is basically everything other than the brain and spinal cord. So it's the cranial nerves, the spinal nerves, and it's also the autonomic nervous system. And the autonomic nervous system, which we talk about as the autonomic nervous system, is broken into two itself. So the autonomic nervous system is split into the sympathetic and parasympathetic systems. And they each serve a compatible, complementary service to your body. Sympathetic is all about fight or flight. If a bear walks through this door, everybody at home is probably going to start recording. Y'all are going to be like, holy shit, there's a bear. Okay? Who thinks that that'll be your first thought? Probably. Then we got some people probably assessing the window situation, how far off the ground it is. That would be flight. Y'all are literally taking flight, like jumping out of the window. You're running away. You see the bear, and you're running away. Flight. The opposite reaction is what? Fight. Some of y'all, and we really don't know until we're really in the moment, some of y'all are going to be like, oh, hell no, that bear is going to be a rug on my floor. And you grab your Apple Pencil, and you just right? That's fight. There's also a third option, it's called like hunker down and like protect. But generally speaking, this is an amped up, ramp up system. Your sympathetic nervous system increases your heart rate. It gets everything pumping and going. It gets you sweating. You're ready to go. You know, like in the instances where someone's trapped under a car and somebody comes over and they're like, let the car up, right? Okay? That's the system amped up and giving you that extra superhuman energy because your body is capable of doing that sometimes. The opposite complementary system, the cool down, is the parasympathetic. This is your rest and digest. Everything kind of halts for a second while you're ramped up, and it's got to basically calm you down. That's the parasympathetic's job, is it calms you down and allows for you to be able to restore homeostasis and create a calm in your body. You want everything to get balanced. That's what homeostasis is there. Okay? Yes.
Like, how does the body determine whether to fight or flight?
That's a good question. So the question was, how does your body determine to fight or flight? It honestly depends on the situation and scenario. So it depends, it's a kind of a knee-jerk reaction. It depends on, do you have the opportunity to run? Okay. It depends on how much stress hormone is being pumped in. It depends on your personality. There's a lot of factors that go into the action that takes place. If you've been in a situation like that before, okay, that might also determine the direction that you go. And there's no way to predict how we think we're going to act, because it's just one of those things where you do. Some people run into danger, some people run away from it, and then some people just are kind of in the middle where they go and find somewhere to hide. Okay, that's not exactly flight. It's a little bit different than that, but those are the three main things that you see in an emergency situation. Okay, good question. Any other questions? You good? All right. So let's talk about the cells of the nervous system. So the cells of the nervous system is what we refer to, the neuron is a nerve cell. We have about 80 billion of these in our brains, and every cell contains information like DNA, information and ability to energize and send signals and messages, and all of that machinery is housed within the cell. Okay? The nerve cells, or the neurons in the brain, are specifically able to send messages through electrical conduction. Okay? Now we're going to talk about how these cells communicate with one another, which is usually chemical. Make sure you're not. And When you're talking about a cell within itself, usually we're talking about electrical conduction. When we have cells talking to each other, usually we're talking about chemical communication, okay? And we'll talk about how a cell does this in the brain, but know that it's all about how the ions in your cells are moving throughout the cell, okay? But we'll get to that in the next section. We have several types of cells in our brain. Many of them are dictated by shape based off of their purpose. Okay, we have lots of types. We have lots of different shapes and lots of different functions. And those shapes are dependent upon the function of that particular cell. So we have motor neurons, sensory neurons, we have interneurons, and we're going to talk about each of those in the next slide. We have different shapes like bipolar, multipolar, and unipolar. And again, you're going to see the shape to be really determined about what the function of that cell is supposed to be. We also have glia, which are all in between all of the neurons to allow for things like being able to clean up any kind of waste that gets produced and also help secure connections together. Another word for glia comes from the word glue, comes from the word glia. It's also referred to as nerve glue. We have about 80 to 100 billion glial cells in our brain. And that is because we need more glial cells to basically glue everything together than we have actual nerve cells. And we're going to talk about those types as we continue on. Any questions so far about the very basics of neurons?
What type of waste do the neurons create?
So the question is what type of waste do neurons create? And it's like any cell. So when cells are consuming energy, okay, there's a process called with ATP, and we'll get into all this later. What happens is those are byproducts. So cells can die, cells can shed basically molecular material, and it will start to accumulate in your brain if you do not have the right kind of waste management system. So just like, your skin cells will die off and, you can actually scratch them off, there are different processes in the brain where it basically creates a kind of waste like that. So it's like a molecular waste. Yeah.
So what would you see in someone if their glias, they weren't cleaning up after those cells?
Yeah, so that's a great question. So what would you see if a person's glial cells weren't doing their job in cleaning waste? So there's a theory right now, okay, that Alzheimer's is one of the causes of Alzheimer's, which is a type of dementia, is actually caused by your brain not clearing the waste properly. Because what happens is the waste starts to accumulate into protein bundles, because part of the waste is made up of protein. So we have these things, we call them plaques and taus, that gunk up the brain, and they're basically taking the space of healthy cells. It's like a dumpster site. And so the idea is that if the brain is not functioning the way it needs to clear the waste, then it'll start to accumulate. And so we see people start to accumulate in certain areas that are responsible for memories and basic functions. And so usually when we see that the waste clearance isn't happening properly, it's not something that's immediate. It might take 20 years to accumulate. But you might see like Lewy body dementia, which again is a type of accumulation in the brain that's causing memory issues and lots of other issues.
Does the waste that you mentioned, does it show up on brain scans?
So the question is does the waste show up on brain scans? And so this is a complicated question because when we scan people's brains with like an MRI or a PET scan, it can't show us a molecular level type of thing. So we can't say, oh, here are all your neurons. It shows us a very kind of macro level picture of the brain. When the waste gets bad enough, yes, you can see it on scans. looks like basically pockets of damage. It looks like atrophy or death. And if you look at a brain of a person who's had Alzheimer's for 40 years, their brain has shrunk. It's very obvious. New issues with waste, you don't really see right off. But if you were to actually take the brain and open it up and slice it, you can locate the proteins through a microscope. But that is where it's microscopic. So it would have to really, really get out of control for you to see it on a scan. And you can, but just not on acute cases. Does that make sense? Any questions about that? Good question. Yes?
If they're not working, is there any way to make the glial cells start working again?
Okay, so the question is if the glial cells aren't working, is there a way to get them to work? So there are a lot of types of glial cells, and so the answer is it depends on what is not working. Generally, though, we can halt some processes from getting worse, but we can't make them do their job and then retroactively heal that process. But again, it depends on the type of cell, it depends on where it is in the brain and what's going on. Generally speaking, we aren't there yet with our treatment and our ability to know what's going on in the brain. Okay, but we're making some advancements, but we still have a long way to go. Okay, most of the time it's just let's just halt where we are and not getting worse or slow it down. Is really the attempt today. Any other questions? Are we good? We have a whole section on Alzheimer's and dementia at the end of the semester. So we'll talk all about disorders and stuff later. And diseases. Okay, if we don't have any questions, we're going to move to the next one. Okay. So here are those types of neurons. So we have the sensory, motor, and interneurons. And these are basic categories of neurons. There are other neurons and types within these. We're just covering the basics today. So a sensory neuron, its job is to get information from your environment to your brain. Okay? So this particular type of neuron is trying to understand where you are in your environment. So it takes the information from like your fingertips, from your eyes, from everything around you, trying to get that information to your brain. And it also works internally as well, and it sends this information to the central nervous system, and we'll talk about that process later. Motor neurons, okay, these are located in this, the CNS. It helps control the contractions of muscles and also the secretion of your glands, for example, sweat. Interneurons are everything sort of in between. These aren't glial cells, these are actual interneurons that help establish connections between the different types of neurons. And again, we'll go into detail about these later, just going to give you the kind of basic categories. And then we have here, and I'm going to show you what these look like too in just a minute. So a multipolar neuron, all of these look different from one another, okay? And generally, when we give you a neuron, we're giving you the easiest picture that we can find, which is usually a motor neuron, of how to identify the parts of a neuron. So a multipolar neuron has a neuron with one axon, okay, and lots of dendrites attached to its body. So this is a multipolar neuron. Here, you see that? You have a soma, which is the body, and one axon attached, okay? And then you have your dendrites here, okay? So that would be an example of a multipolar neuron, and this is our sample example that we usually use to have you identify the parts, because it's the easiest one to know where the cell body is, where the dendrites are, where the axon is, and then where the terminals are at the end. Okay, that's an example of a multipolar neuron. So a bipolar neuron has a neuron with one axon, one dendrite attached to the cell. So a bipolar neuron has one soma, okay, one dendrite here, one dendrite branch, okay. Everybody see that? A unipolar neuron has a neuron with one axon attached to its cell body. Okay, and it divides. I'll show you. This is a unipolar neuron. We have one cell body, and then we have it splitting right here. Okay? And so what's important about the unipolar neuron is that it's branching out to receive sensory information and on one end, and then it's sending information to the synapse. Okay? So one branch is receiving and one branch is sending. This is really important for things like reflexes, because you need a really fast response. The cell in of itself is getting info and sending info. So when you touch something hot, bam, your hand's off it. Okay? So they kind of defy some laws sometimes to get that reflex really quickly reflexed. Because this type of neuron is sending and receiving in of itself. Does that make sense? So it doesn't have to wait for someone else to tell it what to do. It's its own boss. So this unipolar neuron is bossy. Okay? Everybody see how this works? So this is a bipolar neuron, unipolar neuron, another example of a bipolar neuron, and then we have a multipolar neuron here. This is going to be the example that I'm probably going to give you that's the clearest way for you to identify the parts of a neuron. So if you're going to study what a neuron looks like and where the soma is, and which is the cell body, we're going to go through the pieces, where the dendrites are, etc., that would be a good one to label and study. And in fact, if you look on Moodle, you'll see that I've already posted parts of the first activity, which is a coloring sheet on labeling the neuron, except I give it a spin. How many of you have had me for intro? Raise your hand. Okay, all right, at home, give me a thumbs up if you've had me for intro. Y'all seen this before, but guess what? I'm gonna make it harder because y'all are supposed to know more, okay? So you will do the same thing where you're gonna label based off of a word bank that I've given you, but you're gonna add parts to it, okay? So you're gonna label additional parts of the neuron that aren't on the worksheet. That's how I'm leveling up, okay? I'll explain the activity later. It's not due for a little while, but I want you guys to just know it's out there and know that there's parts to that worksheet. So there's the neuron labeling that's you're going to see. There's brain lobe labeling that you'll see. We're going to get that to chapter 3, and then structures, and again, when we get to chapter 3. So I just threw it on there so you could take a look, but it's not due for a little while. Okay. Any questions so far about the types and shapes? You good? Okay. This class is over at 12:15, right? It's 11:30. Okay, so let's go over the parts of the cell. Okay? So hopefully many of you are familiar with this. Okay? Anybody already very comfortable with this material? Am I going too slow? So I've got some people shaking their heads. They're like, no, I don't know. Okay? So the cell body, or the soma, okay, is right here. This is the body of the cell. So the soma or cell body, soma is just, I believe, Greek word for cell body. And it houses all of the machinery of the cell. It contains its DNA, it contains instructions on what it's supposed to do, and all of the processes that keeps the cell alive. Okay? We have the axon hillock. This is the point where the cell meets the axon. Okay? Right here. The axon hillock is the part of the cell where the axon, okay, that long slender part, meets the cell body. That's really important for transmitting this electrical signal right through the cell, okay? So it's important to take the signal from here and then throw it down the axon. The axon is an extension of the cell, and it carries that electrical impulse down the course of this cell. Now, one might think that it's just shooting down in a linear form. It actually does not. Okay? Of course, I don't have my next one. These right here are called nodes of Ranvier. And what happens is the electrical impulse jumps from node to node through the cell to the next end part of the cell. Okay? So it goes through the axon and jumps from node to node. The dendrites receive the impulses. Okay? This is the receiving end from taking information from the cell before it. Dendrites is also comes from the word tree comes from the word dendrite. Okay, so like dendrite, like tree branches, receives information from another cell. That information then will tell the cell what to do. Do you want me to send an electrical impulse? If it gets enough information that says yes, it will then work a magical process, which we'll talk about later, called action potential, and then it'll send that electrical signal through the cell. The synapse is a tiny little gap between one neuron to the next, where information is passed from cell to cell, and the synapse will be here at the end connection, which will be to the next cell. Moving on, and we're going to have a picture of this later. We'll talk about the synapse. The cells are actually not physically touching. There is a little gap, okay, and that's the synapse. Then we have the axon terminal or terminal button, and this is the end of the cell that leads to the synapse. Now, some people like to use the word bouton, terminal bouton. I think that sounds rather pretentious, so I'm just going to call it a terminal button. But if you hear the word bouton, and sometimes a variation in spelling, just know that's also an accepted way to talk about it. Finally, we have dendritic spines, and these are little extensions of the dendrites to basically increase the surface area of the dendrites. So you see these like little bumps? These are spines. What that means is it allows for more connections to other neurons because there's more surface area for it to connect to. The more connections you have, in theory, okay, the more communication that you can have, and the better, the faster you can do things. Okay, so you can actually see in a mouse brain these dendritic spines formulate after being taught a new maze. You can actually see through the learning process these dendritic spines increasing. Guess what happens if you don't use the cells? Those dendritic spines will shrink. If you don't use it, you lose it. Okay? So, any questions about the makeup of a typical neuron? Yes.
You're talking about the gaps.
Yes.
So it seems like there's two places with them. Why is that?
It seems like there's what?
Two places. So the myelin sheath has the gaps, and also the terminal buttons, like the synapses.
Yes.
They're
Okay.
connected by gaps instead of actually touching. Why is that?
Do you mean right here and right here?
Yes.
Okay. So the question is about the gaps and why there seems to be two places where there's gaps. So the gaps, or the nodes of Ranvier, vary depending on how long the axon is and how many times the axon splits. So wherever there is axon, there should be the myelin fatty sheath that's going to cover for better conduction. And if you have like 3 splits in the neuron, you're going to then see each split having additional myelin sheath cover with the nodes of Ranvier in between. Now, I'm going to show you a picture of what the myelin sheath look like, and you'll understand how it's wrapped and why you see those nodes. So I'm going to get to that in just a second, but it's a really good question. And you'll see how it allows for just enough of the axon to be exposed to allow for the signal to jump from node to node. Does that partially answer your question?
Yes.
Okay. So here, and this is just an example of one split. When you look at a true neuron, it's many dendrites and many splits in the axon, depending on the type of cell. Does that make sense? All right. Alrighty. Any other questions? Yes.
For the dendritic spines, do
Yes.
they grow as you use the cell more?
Yes. So the question is, does the dendritic spines grow as you use the cell more? Yes, it does. And we see this process most easily during learning. So when you experience something new, like today, all of your dendritic spines in particular areas of the brain are actually getting bigger to allow for more connections. And then you're actually seeing connections make between additional cells. So that's kind of the process to help establish new information and new ways of doing things. Okay? Everybody good? Everybody good at home? Now throw me a thumbs up if we're all good with the neuron. Great, thank you guys. So here's an example. So this was just an example of one cell to look at how it looks independently of others. This is a more realistic picture of how the cell is connected to other cells to allow for communication. So here, the same cell as before, we have the soma, we have the dendrites, we have the axon, and then the terminal boutons or buttons here. Now, here is another cell. Notice that its axons right here, its terminal buttons are here, and they're connected at various parts on the dendrites and also sometimes on the soma or cell body itself. Honestly, the closer the location of the connection is actually quite important in terms of how it influences the next cell. Not all connections are treated equally, okay? So if you have a connection to a dendritic spine here, and this is sending a message or an impulse here, but if you had a connection directly on the axon hillock, the axon hillock connection actually has more power in the decision to relay, is the cell going to conduct the electrical impulse or not? And then, and we'll talk about this a little bit more in a minute, these impulses actually add up. So if you have 40 connections, and every one of those is telling the cell to fire, or to conduct that electrical signal, then that's going to add up and potentially set it over the limit to then conduct that electrical message. But we'll get more into that later, but just want you to kind of visualize this. So here is another cell, its axon, its terminal buttons, connected to the soma and to the dendrites here. This is the cell. If the impulse, if the threshold is met and the cell is going to fire or send the electrical signal, it's going to send the signal through the axon, okay, to the ends of the terminal buttons that's connecting to the next cell's dendrites and sometimes cell body. So here, in between this connection here, which is not a physical connection, there's a tiny little gap, and what's that gap called? The synapse, or the synaptic cleft. That's going to have a tiny little gap where chemical information will give the next cell instructions on what to do. Okay? So, if the cell fires, the electrical impulse goes through, it's going to then be able to send some message to the next cell to either fire or not. Okay? Any questions about that? Are we good? Yes.
What if it receives conflicting information?
And it does. It will. So the question is, what if it receives conflicting information? Every single time it's going to receive conflicting information. Which one is more? So sometimes it'll send a signal that says, don't fire. And then this other signal says, do fire. And this other says, like, do fire. And the other signal is like, don't fire. Okay? Wherever the signal's being sent, connections. Okay, what power do they have and how many? Okay, that will determine what wins out, fire or no fire. Okay. Any questions so far? We good? Pretty good? Okay. So let's talk about those glial cells, the glue. These are the cells that help provide a warm and inviting environment for our neurons. These are our support cells. They provide nutrients. They allow a buffer from the environment to allow for protection against the different cerebral spinal fluid and whatever else is around it. It also helps to anchor the cells in place. You don't have cells floating around in your head. These help kind of keep everything into a secure network. And it also helps assist in the development of the brain. So the three types that we're going to cover today are astrocytes, oligodendrocytes, and microglia. Okay? So the astrocytes, these are all types of glial cells in different parts of the brain that have different functions. Okay? So the astrocytes are all about the physical support, providing the nutrients, and making sure that the neuron's environment is the right kind of environment to ensure its survival. Making sure that the right electrical environment's in place, that it's not going to kill the cell, basically. Okay? The oligodendrocytes' job is to help form that myelin or fatty sheath in the central nervous system cells. It wraps around the neurons to allow, it wraps around the axons, okay, to allow for better conduction of the electrical signal. Does anybody know what it's called when you have myelin sheath that starts to degrade? Does anybody know what disorder is caused by myelin sheath degrading? Nope, good guess. A lot of people refer to it by an acronym. Anybody at home know?
Is it ALS?
Oh, that's such a good guess. It's not ALS. We got to guess at Parkinson's. It's not Parkinson's.
What's ALS?
It's basically Lou Gehrig's disease. We'll talk about that later. It is a very progressively neurodegenerative disease.
Gotcha.
I'll give you the acronym if you guys can tell me what it is. MS. Multiple sclerosis. Yep. Multiple sclerosis is the disorder that happens when your myelin sheaths start to degrade, which means that those cells cannot conduct electricity properly. And depending on where it occurs in the brain, you'll see the symptoms. So if it occurs in the neurons that are associated with vision, then the person's vision will start to go, and they might get blind spots, or they might get blurry vision. The person's part of the brain where their motor cortex is, where they'll start dropping things and potentially tripping and falling. A lot of times what happens is it's not isolated to necessarily one particular area, and then it sort of kind of takes over different things, and you have lots of different symptoms. You can actually see this on a scan where you see pockets of brain damage, basically, from the cells not being able to work properly, and then some of them are just not going to be able to survive. Okay? Any questions so far about that? And again, we'll talk more about the sort of stuff just kind of while we're coming through. So on the end, the final one is the microglia. And these are the ones who are specifically responsible for clearing out the debris and waste. They're the ones making sure, they're like the dump truck. They're making sure that everything stays nice and clean. Okay? Any questions so far? Are we good? Okay. So right here, this is so what you can do with cells is actually stain them. So you can stain them certain colors so that you can look under a microscope and be able to very easily tell based on the protein composite of the cell. So here, Blue is the nucleus of this neuron, okay? Green is the neuron itself, okay? The red is all the glial material and cells that are kind of holding things together. So as you can see, we have a lot more glial matter and material than we do neurons because that's the glue that's holding everything together. Does that make sense? Just give you a visual on that. So let's talk about the myelin. So myelin, this is that fatty substance that's going to wrap around some axons. And again, I say some because we're going to talk about a lot of different types of cells. And every time you try to establish a rule, there's a new cell or process that breaks the rule. So I try not to give you too many absolutes, like cells only communicate chemically, and some of them don't. Some of them do communicate electrically, some of them do communicate via gas. We'll get into the exceptions later. But generally speaking, the myelin surrounds the axons, and they go around those nodes of Ranvier. Two types of glial cells formulate the myelin sheath, depending on where it is in your body. In the central nervous system, the oligodendrocytes formulate the myelin sheath. In the peripheral nervous system, Schwann cells are the ones that are surrounding the neurons or the cells. So here, the oligodendrocyte here, as you can see, is formulating the myelin sheath. Okay, these are different axons here, and it's actually surrounding the cell. Schwann cell here, you can see wraps around a little bit differently. That's in a peripheral nervous system cell. And here we can see it even better, how it's actually wrapping the cell. So Jared's question here is, you can actually see the soma, or the cell body of the oligodendrocytes, physically wrapping around this myelin as a myelin sheath. Here, so it's wrapping to allow for conduction, and then you can see how it's actually skipping over little bits, which are the nodes of Ranvier, okay? And then you can see here, and we're going to talk about the inside of the axon later, but know that the inside of the axon actually contains microtubules that can allow for, some cases, the synthesis of certain chemicals, the transport of those, and we'll talk about those later. But here is a really nice visual of how it wraps the cell. So here you can see the tiny little gap here, okay, and how it's wrapped the cell. Does everybody see that? That's a nice visual. You guys have probably never seen the myelin sheath in this angle before, right? Okay, because everything is kind of packed in as a network. And so you don't really, we single out cells to be like, this is what the cell looks like. But this is how it looks like in relation to other cells.
So what is this process doing in the body? Like what is the effect of wrapping around that?
So we need so the question is what is the process, the effectiveness in the body? What is it doing? So what this is doing is all the cells in your brain are getting, their axons are getting wrapped with this particular type of glial cell, this oligodendrocyte, to allow for the electrical signal to get passed within the cell itself. So you know when you have like an electrical wire, okay? What do you notice a lot of times with electrical wires?
They're wrapped in like a rubber.
They're wrapped in an insulator, exactly. That's the job of the glial cell. That's the job of the myelin sheath. It's a fatty substance that wraps the axon, just like you see a wire wrapped in an insulating material. It allows for the conduction to go through the wire and, not expose it, because if you expose it, then it can travel anywhere and it can't be a focused electrical signal. So what this does is it allows for that signal to jump from point to point, protect the axon, and allow for really fast speed of conduction. Okay? Yes.
For longer cells, are there like multiple oligodendrocytes just lined up?
Oh yes, yes.
Or is it just one oligodendrocyte that covers it?
No. So what you'll see, and the question was, is it just one oligodendrocyte that's wrapping the cell? You can actually see from up here This would be an example of probably one right here. This is one probably over here. So you're going to see overlap of oligodendrocytes, and you're going to see them throughout the network. So think of it like a fishnet, right? Everything is just kind of on itself. So think of it like 3 fishnets together, and they're all kind of interwoven with each other to provide a network of cells. Okay? Any other questions? Are we good? Okay. Good questions. So another part of this system is something that we refer to as the blood-brain barrier. Now, the blood-brain barrier is basically the bouncer of the brain, okay? So if anybody's ever been to a club and experienced bouncers, their job is to not allow people in clubs, right? So the blood-brain barrier is like, nah, you're not getting in, you're not on the list, okay? Now, this blood-brain barrier is what we refer to as selectively permeable. So it's a barrier between the bloodstream and the brain. Because it's selectively permeable, it allows some things in. So permeable means through. If it was 100% permeable, everybody would get in, and we know that clubs don't work that way. So it's selectively permeable. It only allows people who are on the list to get through, mostly due to size. Okay? These bouncers are very sizist. Okay? The bigger you are, the less likely you're going to get in because you have to fit through the gaps. The smaller you are, the easier it is to get in the blood-brain barrier. Okay? So here you can see that there's capillaries through the system, right? Okay? Capillaries in the brain, part of your the blood networking, right? And here we have these tiny little gaps. These gaps are the selective permeable part of this. These little gaps do allow for certain tiny molecules to get in, okay, and get into the brain and be able to do different things, okay? So like, for example, one molecule that's too big to get into the brain is dopamine. It's huge. It doesn't get to the brain. So if you give somebody a pill and it's pure dopamine and they take it, it'll do nothing. Okay? You'd be like, how do you get people to make more dopamine? Or how do you get people to have more dopamine? You have to give them medications that will make them either make more or change the process, or you can do something like give them tryptophan, which actually is a precursor to serotonin, give them a precursor, the molecule before it gets to dopamine, okay, and trick the brain into making it itself. One thing that is super important to get through the blood-brain barrier is glucose. Your brain uses glucose for energy. So you need that glucose, which is what you get from what you eat, right, your blood sugar. You consume something, goes to the bloodstream, and it's going to go to your brain as fuel. Yes?
Is that where we see problems with diabetes and stuff?
That's a great question. So, yeah, so the question is this is where we see problems with diabetes? Absolutely. If your blood sugar is off, either too high or too low, you can create a problem in the brain, okay? So if your blood sugar dips, you can pass out. If it gets too high, you can go into a coma. Okay? Your brain needs to have the right balance of molecules, ions, electrical molecules, and metabolic substances. Okay? Glucose is very important to balance this out and make sure you have the right amount of energy. Okay? Your blood also contains oxygen. That is also something that your brain uses for energy. So if you go without oxygen to your brain, what happens? What could happen? It could die. Absolutely. This is where we see strokes. Okay? You have a lapse in oxygen to your brain. Your brain does not have fat storage. It cannot It does not have a storage of energy. It uses energy as it gets it. So if you're using a part of the brain, blood gets pumped to that area to provide it oxygen and glucose to use as energy. Okay? And when you remove that, then that part of the brain will die. So, why do we have a blood-brain barrier? One is we want to prevent certain substances, mostly toxic substances, from getting in the brain. Interestingly, okay, pain relievers like Tylenol, okay, do not get to your brain. They work on the peripheral nervous system to help relieve pain. Other drugs, like opiates, can pass through the blood-brain barrier and actually impact brain activity, which is why they work so well. Okay? So one, blood-brain barrier prevents toxic substances. Two, it helps to balance the fluid. Because if those gaps are too big, then your blood's gonna literally seep out. Not good for your brain or body. Okay? You don't want any of your cerebral spinal fluid seeping in. Also not good for the brain. Okay? So it helps keep things in and help keep things out. There is an interesting area in the brain that is not part, that does not abide by the blood-brain barrier rules. This is the area of postrema, located right here below the cerebellum on the brainstem. Anybody want to know why we should have an area of our brain where the blood-brain barrier does not influence it? Jarrett?
Because the stuff that's going into your brain is also in your body.
Okay.
So I guess it says here that it controls vomiting. So if there's
Yeah.
poison in your body, your brain needs to know that.
Very good. Okay, so the answer was there are things that are going through your body, and there's an area that needs to know if it's toxic or not to expel it. I know I have summed it up, but that's basically what you said, right? That is the purpose of the areal postrema. It controls vomiting. If you consume something that's toxic, it is going to immediately trigger the uh-oh, throw it up. Let's get rid of it. Okay? This can be good and bad. There are some things that can actually quiet the area postrema. So for example, if you consume too much alcohol, your brain is going to allow that to go through the blood-brain barrier. Area postrema says, no. Expel, expel, expel. You're puking your guts out, right? To save you, hold on one second, from alcohol poisoning, okay? Let's say you take another type of chemical that turns off the area of postrema. Does anybody want to venture a guess what you can take that will turn this off? What's that?
Like medicine that controls.
I'm saying even simpler than that.
Oh, okay. I don't know.
What do you guys think? What's something that you can consume or take or smoke or marijuana? If you consume marijuana, it'll turn off the area postrema, which is why it's so dangerous to ingest or smoke or whatever marijuana and alcohol together. Because if you are consuming too much alcohol, your body's not getting the signal to puke it all up. And the question on chat was, could allergies be registered as a toxin? So allergies actually aren't necessarily a toxin. It's your body's response to something natural that's triggering what feels like a toxin. But it's not necessarily the same thing. Interesting question, though. It's the same sort of process. I saw a hand. Did I answer your question?
So during pregnancy, does it become more sensitive, like to protect the fetus from toxins? Is that why there's more nausea?
So that's a really good question. So the question is during pregnancy, do you see the system being more sensitive to protect the body? You do. You actually see that women who are pregnant tend to get turned off from certain things like smells, a lot of times meat and fish, and certain things that could potentially be toxic. It ramps the system up. And what's responsible for that is hormones. So the hormones play into this. And so, like, for example, if you eat raw fish, a lot of people don't like the smell of raw fish because, you know, they associate it with undercooked food. A lot of women, myself included, in fact, early in pregnancy, with one of them, I walked through a fish market and almost puked my guts out because I was like, oh my God, I can't. And I love sushi and I love all that stuff. I could not eat it when pregnant. But yeah, it's a protective factor. It's your body kind of ramped up to be extra sensitive to protect your body and the fetus as well. Yes.
Does that mean that the body registers those types of foods as like not necessarily toxic but harmful to the
Oh,
fetus?
sure. Have you ever smelled rotten meat before?
I don't know. Probably yes.
I feel like you probably.
Yeah.
Okay, so let me ask you guys this. So the question was, does that mean that some things in your body responds to because it's dangerous for you, right?
Yeah.
Have you ever smelled a rotten animal?
Yeah.
Do you have any desire to go and cook that animal and eat it?
No.
No. We are repulsed by things that can hurt us, okay? Because they can hurt us, okay? That's why if you smell something and it doesn't smell right, it turns you off. It's your body's way of protecting you, your brain's way of saying, hey, dummy, don't eat that because you're going to get sick and die. It's an evolutionary mechanism, basically.
So if you had like the fish, if you had gotten a fish from the fish market, do you think it would have, was that your body telling you that it would have upset your stomach?
Oh, so the question was, if I would have gotten fish from the fish market, would it have potentially upset myself?
Yeah.
Probably. I couldn't even get to the point where I would even hold one because I was like, ugh. I was like booking it. I was in Atlanta and I was like, I was in a market and I was like, I've got to get out of here. I couldn't even get up to the counter. I was like, I'm just watching straight through this. So probably. I probably wouldn't even be able to physically do that. Just that's how repulsed I was. Good questions though. Any other questions about the blood-brain barrier? I think we'll probably do 1 or 2 more slides and then we'll be done for today. Are we good? Questions? No? Good questions. Okay, so hold on 1 second. I'm going to skip ahead real fast and see where we're at. We got 2 more hours. We're close to being done. Okay. All right. I thought I had like 5 more slides. Okay. This is great. This is going to lead us so well into next time. Okay. So I've already mentioned this, okay? But I really want to send the point home because this is something that you really need to understand. I'm going to say this and then later I'm going to lie and I'm going to pretend like I never said this, okay? All the transmission within the neuron itself is electrical. It's electrical conduction. The impulse travels from the axon hillock. Remember, the axon hillock is where the cell body meets the axon. It's going to just jump its way down through the nodes of Ranvier to the end of that neuron. That is action potential. And we will cover that next time. Between the neurons, after the signal is sent, and it's sending a message to the next neuron, usually is chemical, okay? These are neurotransmitters. This is the chemical signal that gets sent to the next stage. Anybody want to throw out a few neurotransmitters? Serotonin. Dopamine. Melatonin is tricky, it's a hormone. ACH, acetylcholine. These are all types of neurotransmitters, okay? Technically, a hormone is a neurotransmitter that's outside of the central nervous system, but we'll get into the specifics later. That's kind of like that, okay? We transmit these chemicals through that synaptic cleft, the little gap, okay? And that information is going to tell the cell what to do and how to do it. Sometimes the information gets translated and sometimes it doesn't, and we'll explain why later. But as everybody see here, within a cell itself is electrical, between cells is chemical. Okay? Now we've got a few cells that defy our laws. So just know that we'll talk about those later. But if I ask you on a test between the two, that's what I'm asking. So here we have one cell giving a bunch of messages to the cell body, okay, and that's going to add up, and the cell the cell body is going to process that information. Then certain electrical activity is going to happen, and we're going to talk about that next time. Through the axon hillock, the decision is made. Fire, no fire. Ones and zeros. That's all we got, guys. Ones and zeros. Either you fire and you do it all the way, or you don't fire and you're done. Okay? Fires an electrical impulse. Boom, 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 boom. Oh, shoot. I was too excited. Sorry. And then boom. Now we're at the connection at the synapse. It releases a chemical to the next cell. Maybe it's dopamine, maybe it's acetylcholine, maybe it's norepinephrine, who knows? I don't know. I'm making this example as I go. And then that information will then get sent to here, telling the cell this way, what do you want to do? Fire, no fire. And then that information, and it just keeps going forever and on. Okay? So, does anybody have any questions about what we talked about today? Yes.
What's the end result of it sending things back?
What is the end result of all of this? Everything. Everything that we do. Okay? So the information that gets sent tells it the electrical impulse. Do I release this chemical that causes me to do this thing? Everything stems back to the chemicals. The chemicals, if you've got a bunch of dopamine, can make you feel super happy. Maybe it's telling you don't release any dopamine, and this message goes to this one and this one and this one, and all the cells that are responsible for releasing dopamine to the next cell, they shut off. Now you're sad. That's a super simple way to explain it, but that's pretty much how it works. All the communication happens. All the chemicals will tell you how you feel, what you're doing, what decisions you make, what hand you're going to move. It's literally all stems back to this molecular process. It's fascinating. This is happening all in your head at once.
I feel like that's
Trillions of
really deep.
What's that?
I feel like it goes way deeper too.
It goes so way deeper.
Yeah, it's like
I'm super
it's
simplifying
really
it.
hard
Yeah.
to grasp.
Just so that you guys can understand the basics to then get to the bigger stuff.
Yeah.
We'll talk about the parts of the brain. When these cells are in this part, this is what you do. You know what I mean? But this is just the cellular foundation. At home, give me a thumbs up if you are good and no questions. Great. Okay, I'm going to end the recording. Hold on one second. I got a question. Do SSRIs change the chemicals exchange or just the way the chemicals are received? This is a super fantastic question. We got 2 minutes left. I'm going to explain it real quick. An SSRI is a selective serotonin reuptake inhibitor. It stops the reuptake absorption. So what happens is you push more serotonin naturally in the cleft. So you have to suck it up by the next cell. So it's changing the nature of the process to make your brain accept more serotonin. You're not actually making more, you're just accepting more. So it changes the way that the next cell takes it in. We're going to talk about this later. Super fantastic question. We'll get to that next class with the neurotransmitters. I'm going to end my recording here. You can stay on.